Okay, good morning everyone. Good morning to our online audience. Hi, Board of Judges, colleagues at the National of Science and Technology Philippines, friends, ladies and gentlemen, a pleasant day to all of us. I am Alan Patrick Macabeo and I will be your Master of Ceremony and moderator for today. We are here to witness the paper presentation of what I call the papers of the rising stars of Philippine science and technology, and they are also at the same time the finalists for the NAST Talent Search for Young Scientists 2020, where 5 finalists from different universities will present their papers to vie for the award. I just heard this morning that the coveted prize, which is the first prize, is 100,000 pesos. So, good luck, guys. We are here live at the Philippine Science Heritage Building Auditorium of the DOST. And because of the pandemic, we are not allowed to organize a big event like we usually do. The 5 finalists are here physically with us while our board of judges are available via Zoom. To formally welcome us to this occasion, National Scientist Raul V. Pabella of the Social Sciences Division of NAST and Chair of the Board of Judges will give us his welcome remarks. He will likewise introduce the member of the Board of Judges.
Good morning, everyone. On behalf of the Academy, I'm delighted to welcome you all to the paper presentation of finalists for the 2020 NAST Talent Search for Young Scientists. The NAST Talent Search for Young Scientists is a project of the Academy to encourage young people to pursue careers in science, and this project started in 1991. Recipients of the award have become successful in their careers and are already distinguished members of distinguished professional and scientific organizations, as well as of NAST. We are conducting these proceedings virtually for the first time for obvious reasons. Remember that this award is as much for work already done as for work still to be done. I would like to congratulate you all for making it among the 5 finalists, who are already all winners. Each of you is already a NTSYS awardee. It is an honor that you carry with you for life. So vanish the nerves and enjoy the rare privilege. This exercise is just being done to get the rankings. The finalists were selected among several nominees based on the following criteria: involvement with scientific undertakings, 10%, other research projects undertaken, 10%, knowledge and mastery of the science and the application of scientific methods and principles, 50%, and contribution towards national development or advancement of science and technology, 30%. Final judging for this award will be 50% for the interview and 50% for the quality of the paper presented. You are entitled to cast awards and plaques. The grand prize winner is entitled to a research grant worth 500,000 from DOS, and of course a cast award of 100,000. The members of the board are eminent scientists. They are academician Gisela Concepcion. She's recognized for her superior contributions and accomplishments in the field of marine natural products, biochemistry, and biomedical sciences. She's a team leader, but also a member of various international research groups to discover novel marine compounds with anti-cancer, antibacterial, and anti-tuberculosis and anti-inflammatory immunosuppressant properties. Academician Concepcion is a member of an international consortium that recently discovered and announced the compound called boholamide, simply because it was discovered, the shell was discovered in Bohol, was found in Bohol. Academician Jose Maria Balmaceda is a mathematician who has made signal contributions to group theory, representation theory, and algebraic combinatorics. Mathematics, as you know, is like New York City of song. And I quote, if you can make it there, you can make it anywhere. Academician William Bill Torres is a former president of Mosaic Communications and an active participant in various government and non-government educational programs concerning applications of information technology. Academician Carmencita D. Padilla is instrumental in bringing genetic technology to the Philippines through the development of clinical genetic service at the Philippine General Hospital. She is responsible for the Newborn Screening Act of 2004 or the Republic Act 9288. Integrating newborn screening in the health care delivery system and the Republic Act 10747 or the Rare Disease Act. Academician Ruben Villarreal, former Chancellor of UP Los Baños, is recognized for his outstanding contributions not only to the Philippine socio-economic development, but as well to the global problem of poverty and hunger. He was instrumental in the varietal improvement of tomatoes, sweet potatoes, cabbage, cauliflower, watermelon, etc., and to the development of the vegetable industry in the Philippines and Southeast Asia. Undersecretary Renato Solidum is Undersecretary for Scientific and Technical Services, officer in charge of Fieldwork, Office of the Director. We hope by these awards that young scientists and researchers continue to be inspired to conduct outstanding researches in all fields, especially now that we face the COVID-19 pandemic. Personally, dare I dream that one day, one of our winners will bag one of the prestigious global awards, perhaps a Nobel. I would like to extend my sincerest appreciation to the finalists and viewers for sharing your time with us. I wish you all a good day ahead. Thank you.
Thank you very much, National Scientist Fabella. Before we proceed, we would like to invite our viewers to follow us on all our social media accounts. For Facebook, you may like us on www.facebook.com/NAST facebook.com/NASTTHL. On Instagram and on Twitter, please follow us on @NASTTHL. We now come to the much awaited portion of our program, the presentation proper. We would like to remind the paper presenters that you are only given 15 minutes to deliver your presentation. Please pay attention to our timer at the front. The question and answer portion will follow after all the finalists have presented their entries. Our first paper presenter is Mr. Darlon Vilantican. He is a homegrown talent, obtaining his Bachelor of Science and Master of Science all from the University of the Philippines in Los Baños. His research entry discussed gene markers that can facilitate the development of varieties with resilience to climate change, resistance to pests and diseases, and improved oil yield and quality. Let's give a virtual applause to Mr. Darlon Vilantican as he presents his paper on de novo genome sequence assembly of dwarf coconut, Cocos nucifera Lee, Katigan the green dwarf. Provides insights into genomic variation between coconut types and related palm species.
Hello. So thank you very much, Dr. Alan, for the wonderful introduction. So before anything else, I'm Darren Atikan from the Institute of Plant Breeding, College of Agriculture and Food Science, University of Philippines, Los Baños. And I'm here to present my paper, the recently published paper at Genes, Genetics, and Genomes, the GG Journal, the Genetic Science of America, entitled The Novel Genome Sequence Assembly. So there are some technical difficulties here for my slides. Here. So the title of my talk is our research published at the Genetic Society of America, Journal of GP, the novel genome sequence assembly of dwarf coconut genome, provides insights to the genomic variation between coconut types and related palm species. So first and foremost, let me introduce or give you a brief background on the Philippine coconut industry. So the Philippines is actually a major coconut producer, contributing to the 24% of the world's production. And it's actually the coconut is a billion-dollar industry with 45% share in the global coconut oil market. And it's actually a largest contributor to our gross value added, or the GVA, to agriculture, and provides source of livelihood to about 3.5 billion farmers that rely on coconut as a source of. So the challenges, however, this opportunity in coconut industry has several challenges. First, our coconut farmers are still considered as the risk. And our coconut yield in the Philippines is actually about 40 to 45 nuts per tree annually, as compared to the other countries like India and Brazil. And of course, in terms of production, we are formally the #1 coconut producer in the world. But now we are just dropped to the number. So there are several factors that have been contributed to this situation. First, our current coconut varieties planted are low yielding in terms of their genetic. Second, our coconut plantation in the most of our coconut plantations in the Philippines are composed of senile coconut population. Plus, the occurrences of pests and diseases and the occurrences of natural calamities like the coconut scale insect and the devastation of the typhoon Hainan. So to address these threats, we must first be familiarized with the coconut biology and its inherent genetic diversity. So coconut is actually a diploid species with a large and complex genome. So it's the only recorded species under the genus Cocos, and it has two major types. We have the tall coconut and the dwarf coconut. So the tall coconut is primarily late fruit bearing with high heterozygosity. As compared to your dwarf coconut, they are early fruit bearing and high homozygosity. So we are familiar with the several or diverse uses of coconut in the Philippines. But well, we are not yet too familiar that the Philippines is actually rich and blessed with very high degree of genetic diversity. So with this genetic diversity, there's a boundless opportunity in order to improve further our coconut varieties in the Philippines. So understanding the biology and harnessing the coconut diversity could address the challenges in the Philippine coconut industry. However, this is not straightforward as we need major tools because to breed coconut is really quite a bit challenging due to its long breeding cycle. So we need some tool here. So we have here the whole genome sequencing. So whole genome sequencing is actually technology that's used to sequence most of our popular crop species like rice, corn, tomato, banana, and oil palm. And this actually revolutionized the plant varietal improvement programs for these crops. So we have here the dwarf coconut genome, the impact of the dwarf coconut genome to the Philippine coconut industry. So we can identify economically important genes as well as develop genome-wide molecular marker resource for marker-assisted plant breeding. Thus, we can develop varieties with resistance to climate change, resistance to pests and diseases, and has improved oil yield. So the major objective of this research is to characterize gene units in coconut and maximize its utility of this genome information for molecular marker development for plant breeding applications. So for the overall methodology, we use the Katikan green dwarf variety because the Katikan green dwarf variety is actually characterized with high homozygosity and genome simplicity. So the Katikan dwarf variety is a popular maternal parent in the hybrid breeding programs of the Philippine coconut variety. So we extracted DNA from this palm and sent it for outsourced next generation sequencing using three sequencing technologies. We have the illuminamizing sequencing, back value sequencing, and Doppler Chicago sequencing. Then we have the high throughput data. Next. So after we have the high throughput data, we perform bioinformatics analysis or computational tools. So first, we have the genome sequencing from the different sequencing platforms and did some genome assembly using hybrid approach. But the genome is a bit, it has some mismatches or base call errors. So we further polish it using TB jelly and Taiwan. However, after this, we have now developed genome-wide SSR markers and sleep markers. So in order to predict the collection of genes in coconut, we further mask the genome for its repeat elements. And now we have the maker pipeline to do the job. So the maker pipeline requires GeneMark, Augustus, and SNAP in order to predict it accurately and efficiently. So now we have the whole gene models for the coconut. And this is fractionally annotated, and we further filtered it for economically important traits. For example, the resistance for pests and diseases, the genes responsible for drought response, and the genes that are responsible for the biosynthesis of oil. Next. So for the major highlights of this research, first, we have successfully assembled and annotated a dwarf coconut genome which has high coverage, has high contiguous genome sequence, and has high quality. And these three factors here are required in order to predict Nordic models. So next we have here forward. So we have here a collection of genes in coconut for around 35,000 genes. So we further classify it. We're also interested with with several elements, not just the functional genes in coconut. We are also interested with other elements like the long transposable repeats in coconut. Sorry po, yung pong figure. Forward po. Yeah. So the long transposable repeats were able to predict the age of the transposons or the jumping genes in coconut and observe a rapid burst of the LTRs in coconut 400,000 years ago. So this event actually coincides with the Earth's transition from glacial or ice age into the interglacial period called the termination V, or it happened 430,000 years ago. So thus, selection pressure imposed by the harsh changes in the environment shaped the coconut gene diversity, including the composition after two decades. Next. So, from some evolutionary and comparative analysis, wherein we predicted 3 rounds of shared whole genome duplication events among the member of the palm family or the Ericaceae. So there are 4 distinct peaks we detected for Katigan dwarf versus the tall coconut, the Katigan dwarf versus date palm, and the Katigan dwarf versus NTSYS. However, when we align the coconut Katigan dwarf against the banana or the musa, we only detected 2 peaks. Thus, we proposed that the Ericaceae or the palm family shares a common WGD beta and gamma events with the musa lineage or the family of banana cinnabaca, but independent WGD alpha event. So with this, we predict that there is a common polyploid ancestor for our coconut that we have here. And that polyploid species is just a little bit unstable, so it needs to be converted again to diploid. So during the diploidization event, we identified some transpositions and translocation inversions with the chromosomal segments of coconut against oil palm. So here you can find some duplications and inversions. Aside from the evolutionary analysis, we also performed the fishing out of the important or economically important genes in coconut. First, the resistance gene analog. So this is for the resistance against pests and diseases. So we classified it based on its function, and these are the composition of the RGNs. At the same time, we also classify it based on its genetic or similarity or sequence diversity, and we found this 3, the phylogenetic. Thus, the functional and sequence diversity of RGAs will provide a framework for gene pyramiding approaches in coconut resistance breeding. Next. We are also interested with the genes involved in the drought tolerance against the drought response of coconut. So we found, based on homology approaches, that the ABC40 drought response genes has the highest number of homologs in the coconut genome, and the drought response or the overall drought response of coconut are involved in intercellular signal transduction, redox reaction, and abscisic acid signaling pathway. Next would be the identification of genes involved in the coconut oil biosynthesis. So making the coconut as the billion-dollar industry. So the gene sequence for alpha and pyruvate dehydrogenase E1 can be found as a single copy in coconut, and we found 3 homologous genes of phospholipidase D alpha-1 like protein in the coconut genome. So there are 3 homologs. So, but what we are most interested with is the alpha gene, because alpha gene is what makes coconut unique as compared to alpha. So we found that the coconut alpha gene has a shorter gene sequence as compared to your alpha due to the deletion of a certain segment of the supposedly intron relative to alpha. So actually, based on this genome information, we were able to publish another paper and recognized as one of the 2020 scientific awards. So next. Now, for the application in actual plant varietal improvement programs, we were able to develop SSR markers consisting of 7,139 SSR markers and 53,500 tree snip loci. So the SSR markers can now be used for assessment of genetic diversity of our diverse collection of coconut germplasm, and also for this. But actually, the most revolutionary result here would be the identification of variants, because this will actually open the opportunities in order to improve our coconut varietal improvement programs in coconut, because we can now perform genotyping by sequencing, targeting this SNP loci towards genomic selection, genome-wide association studies, and high-density genetic mapping. Next. To summarize this talk, the coconut Katigan dwarf genome was assembled and annotated using combination of 3 sequencing platforms. We detected 3 rounds of whole genome sequencing, whole genome duplication events in Aracasi or the palms, and the genes associated with several economically important traits in coconut were identified. Furthermore, we have developed genome-wide DNA markers for coconut tree. So I hope that our result in this paper would not only just a paper alone, but also to alleviate the current situation of our coconut farmers. And I do believe that as a genomics, as a person who works with genomics, the technology advancement in science and technology could awaken the sleeping giant in the Philippine economy. And that is the sector of agriculture. And that will be my goal as a young scientist. Thank you very much and have a nice day.
Okay. Right. So our second paper presenter is Dr. Lurico de los Santos Lapitan Jr. He is a principal investigator of the Advanced and Nanomaterials Group of the Research Center for the Natural and Applied Sciences and a faculty member at the Department of Chemical Engineering at the University of Santo Tomas. He obtained his on our screens Dr. Lurico de los Santos Lapitan Jr. as he presents his paper on combining magnetic nanoparticle capture and polyenzyme nanobed amplification for ultra-sensitive detection and discrimination of DNA single nucleotide polymerase. Dr. Lapitan, please.
Hello, good morning, everyone, and to the esteemed board of judges via Zoom. So first, I would like to apologize if there's a bit of change in the title. Okay, so again, good morning, and this time I'm happy to share with you, okay, one of the PhD projects that I did. Okay, so this is all about the ultra-sensitive DNA sensing, specifically cancer-related DNA sensing. Okay, so today we are facing a lot of healthcare challenges. And as scientists, the big question is, how do we solve this problem? So hopefully we will think about this problem and hopefully we'll try to have an answer at the end of this presentation. Okay, so all right, so the global impact of cancer burden is 18.1 million cases. And in 2018, we have 9.6 million deaths. In the Philippines, okay, the cancer cases is steadily increasing, and we have around 141,000 new cases in 2018. Now, this pie chart here tells us the different number of cancer cases that affects Filipinos. Okay, so this is the grim reality of cancer, and we have to remember that cancer will always be here. It will be a major global challenge. So, as scientists, how do we go about this? What is the best strategy? So the best strategy is early diagnosis. Okay, so this is the best way to defeat cancer, and not only cancer, any types of illnesses, any types of viral illnesses or the pandemic, for example. Okay, so why is early diagnosis very important? For example, for a small tumor and for early HIV infection, disease-related markers are present in the blood at very low concentrations. And these concentrations are actually undetected by our conventional diagnostic tools. So as scientists, we should be able to develop detection methods for devastating diseases. And secondly, we have to remember, okay, to improve the sensitivity of our diagnostic tools. Okay, so at this point, I would like to include, okay, the realm of magnetic nanoparticles. These are very exciting materials to work with in developing diagnostic tools. Okay, so multivalent magnetic nanoparticles are actually made when we combine magnetic nanoparticles with biomolecules and small molecules. And we need the knowledge of chemistry in order for us to create these functionalized materials. In fact, okay, multivalent magnetic nanoparticles are actually used to destroy, to kill cancerous cells by hyperthermia, or we can use magnetic nanoparticles to actually put some drugs on it and deliver it to the tumors. Okay, so magnetic nanoparticles, we can also use them to capture the target of interest in the solution and to get them or remove them from the solution. That is magnetic separation. And the last major application of magnetic nanoparticles is for sensor development. Okay, example in magnetic resonance imaging, and more specifically, which is the topic of my talk this morning, is the use of magnetic nanoparticles to create biosensors to diagnose certain diseases. Okay, so let's now focus on single nucleotide polymorphisms or SNPs. These are the most common type of genetic variations among individuals. Now, SNPs are actually largely benign. However, the SNPs may tell us or may predict, okay, why people, for example, have certain different responses to drugs, why people have different responses to toxins, and why people have different predispositions to developing cancer and other complex diseases. Okay, now we have actually a surge of interest. Actually, it is an exciting area in biosensor of developing new methods, okay, to detect SNPs, specifically, for example, cancer-related SNPs. For example, if you have a single base mutation in the KRAS gene, that is when the C is mutated to T or when C is mutated to A. Okay, these are actually related to certain types of cancer. Okay, so to achieve this goal of developing new diagnostic tools for cancer-related SNPs, okay, I use a technique that I developed, which is called the magnetic nanoparticle enzyme sandwich sensor. Okay, so enzyme sandwich sensor, I will discuss what this one is in a while. So first I will tell you first what are the components of this sensor. Okay, so there are three components of this sensor. First, we have here the magnetic nanoparticle. These are the black circles that you see here, and these magnetic nanoparticles are actually stabilized by a polymer, which I will discuss in a while. What is this polymer? Okay, so the second component for this strategy is the use of enzymes. Now, why enzymes? So enzymes are actually excellent signal amplifiers. Okay, so in this study, I used a specific enzyme called neutramidine HRP. So obviously, this is just a representation just to make things look nice. Okay, so and thirdly, we also have some DNA probes. Okay, so the DNA probes are the following. So you have here the captured DNA, which is actually linked to the magnetic nanoparticle. We have here the target DNA, which is the analyte of interest. This is what we want to detect, something like that, and to quantify. And of course, the last one is the signal DNA. Now, take note for the signal DNA, you have here a biotin group at the end. So this biotin binds actually to the enzyme. Okay, so these are the components of the sensor. Now the next thing is, how does the sensor work? Okay, so the sensor works like this. So you have here, for example, the magnetic capture DNA, and the DNA part of this one, okay, is linked to your target DNA. Now we have to take note that the sequence of this, the half of the sequence of this target DNA is complementary to this DNA. Okay, and then we have here the signal DNA. Okay, so meaning, okay, the capture DNA, which is the blue one here, is binds only with the, let's say, signal DNA only in the presence of the target DNA. Right. So the next step is to incubate this with the enzyme. All right. So in this figure, okay, you would see that the DNA portion is actually sandwiched between the magnetic nanoparticle and the enzyme. That is why I call it the magnetic nanoparticle enzyme sandwich sensor. Okay? Right. So the exciting thing happens now. Okay. So the exciting thing is, if we have, for example, a colorless substance, which is amplex red. Now, in the presence of the enzyme catalyzes the amplex red to a strongly colored material and a strongly fluorescing material called resorufine. Now, the intensity of fluorescence, okay, of resorufine is directly correlated to the concentration of the target DNA. So in that way, we can do the biosensing or the quantification of our target DNA. Now, the resorufine, okay, can be monitored, or the intensity of the resorufine can be monitored, okay, using a simple plate reader, which is actually available, I think, in most clinical laboratories. Okay, now, to improve the detection limit, okay, to make it more sensitive, for example, okay, instead of using only one enzyme to bind with the biotin, I have made a novel strategy, a powerful, okay, signal amplification strategy in which I used a polymer bead, and I tagged this or linked this one with multiple copies of the enzyme. So the idea here is that the multiple copies of the enzyme will convert, okay, more of the amplex red to, oops, sorry, there's a technical problem. Sorry for that one. Okay, so going back, okay, so the multiple copies of the enzyme on the polymer bead, okay, will convert a lot of amplex lead to resorufine, leading to a higher intensity of fluorescence, even at low concentrations of the target DNA. That is one way to improve sensitivity in biosensing. Okay, so as I've mentioned earlier, okay, the magnetic nanoparticle was stabilized with the polymer. So I have synthesized this polymer, okay? So I will not go on with all the steps that in the synthesis of this polymer, but I just want to pinpoint that this polymer contains the azide group. Contains the N3 or the azide group. Now, why is this important, okay? So the azide group, I use the azide group to link or attach, okay, the capture DNA to the magnetic nanoparticle via copper flick chemistry approach. Okay, so the next thing is for me now, okay, I have my polymer. So the next thing for me now is to use this polymer to coat my magnetic nanoparticles with the aim of dispersing them in aqueous medium. Okay, so this is the slide that tells us, okay, that after several trials of experiments, I figured out that the 1:10 weight ratio of the magnetic nanoparticle to polymer weight gave me, okay, the highest encapsulation efficiency. I've also monitored, okay, the hydrodynamic sizes of these encapsulated polymers, and for 1:10, it gave me a hydrodynamic size of 223 nanometers. So what does this results tell us? Simple, okay? The results tells us that the polymer that I synthesized was able to encapsulate the magnetic nanoparticle and disperse them successfully in water. Okay? So the next thing now is obviously to find out how stable is our polymer encapsulated magnetic nanoparticles. So I dispersed my polymer encapsulated magnetic nanoparticles in solutions of different pH, different temperatures, and different solutions containing different concentrations of the salt. So overall, I will just summarize, okay, the polymer encapsulated magnetic nanoparticles are stable at a wide range of pH over a wide temperature range, and this one is also stable at solutions of different salt concentrations. Okay, so we are done with our material. So the next thing for us now is to do the biosensing. Okay, so biosensing was done in A, this is in pure buffer, and in B, this is a buffer containing 10% human plasma. So why did I add 10% human plasma? That is simply to mimic clinical assay conditions. Okay, just to summarize the results, okay, the target one S1, which is the perfect match, which means the red one, that this one is the target DNA, is perfect match or perfect complementary with the captured DNA, which is the blue, and the signal DNA, which is the green. And this one is expected to give a highest fluorescence reading. And indeed, in my study, it gave the highest fluorescence signal, okay, T1S1. Now, the T3S3 or target 3 signal 1 or target 2 signal 1, which contains a mismatch. For example, when C was mutated to A or C was changed to T, for example, the signal is almost similar to the blank or the without target. So overall, what does this tell us? The biosensor is actually highly specific. It is highly discriminatory, highly specific. All right, now in order to improve the sensitivity of the biosensor, as I have mentioned previously, I designed a polymer bead and I linked this one with multiple copies of the enzyme. Okay, and I did the biosensing. Right, let's look at what's happening here. So if we look at this graph here, okay, the rate of fluorescence production is increasing linearly with an increase in DNA concentration. Okay, and that is a good news. Okay, now the next thing is to analyze this. So analysis of this graph gave me a linear relationship of 0.966 and a wide linear dynamic range of 6 orders of magnitude. In biosensing, it is nice actually if we have a wide order of linear range. Okay, so upon computing the limit of detection or how, or what concentration the smallest concentration that my biosensor can detect, okay, the limit of detection was computed to be 1.6 attomolar, an excellent concentration of 1.6 attomolar. Now the next question is, okay, how do I fare with the global biosensing platform? How good is my biosensor that I developed? So I have summarized here, okay, the world's best biosensors. And I'm happy to tell you that the biosensor that I developed is at par, okay, with them or even the best in terms of limit of detection. Okay, so in summary, okay, in this presentation, I was able to show 3 things. The first is that, okay, the encapsulation of hydrophobic magnetic nanoparticles using the polymer that I synthesized, okay, provided a stable and well-dispersed magnetic nanoparticle. And this polymer that I designed as well is a convenient way to attach our captured DNA via copper-free click chemistry. Okay, the second important thing is that, okay, I developed this new biosensing strategy with excellent SNP discrimination or a biosensing strategy with that is very specific using these different steps that I have explained earlier. Okay, and the other highlight of this study is that using a simple strategy wherein you have a polymer bead and you tag this one or link this one with multiple copies of the enzyme, you can actually detect DNA concentrations down to the 1.6 attomolar. That is PCR free. Okay? Right. Okay. So overall, in my study, I developed a strategy to detect DNA for cancer diagnosis. However, this study can also be translated to the diagnosis of other diseases, like say, for example, viral diseases. Okay, so ultimately, what I'm trying to say here is that early diagnosis is very important. Okay, early diagnosis is important because we can easily pick the occurrence of diseases. And doing that one, we can actually curb the spread of diseases and improve the lives of our fellow men. And who knows, we can also, save our economy from the downfall of, let's say, a pandemic, something like that. Okay, now I'm also happy to share that in this single project, I was able to communicate the results in 3 high impact factor journals from the Royal Society of Chemistry and in Elsevier. Okay, now before I stop this presentation, I would like to thank everyone who is listening now. Hello to my friends in Facebook Live. Okay, hello everyone. Okay, so acknowledgement goes obviously to the Zhao group, okay, my group in UK where I did this project, and to the University of Leeds and to the University of Santo Tomas Faculty of Engineering for allowing me to pursue my studies. Again, thank you very much. For those who have listened, thank you very much to the panel of judges via Zoom. Hello po mga ma'am and sir. Maraming salamat po.
Okay, thank you, Dr. Lapitan. Right, so our third presenter is Dr. Lynn Marie de Jesus, de Juan, sorry. Corpus obtained her PhD in Material Science and Engineering from Hokkaido University, Japan in 2018. She is also a principal investigator of the Engineering Science cluster at the Research Center for the Natural Sciences. Her research entry demonstrates her talent to probe new and important possibilities about batteries. Let us all welcome on our screens Dr. Lynn Juan Corpus as she presents her study on rechargeable batteries utilizing one-dimensional vanadium-based electrode, past, present, and future. Dr. Corpus?
Good morning, everyone, and good morning to the panel of judges. I'm Lynn Marie de Juan Corpus, a PhD in Material Science and Engineering, and I'm here to present our research entitled Rechargeable Batteries Utilizing 1D Vanadium-Based Electrode, Past, Present, and Future. So this research is a collaboration among UST, Hokkaido University, Chulalongkorn University, Chungyuan Christian University, and Nanolabs LRC Company Limited. So lithium-ion battery dominates the battery market due to its wide range of application. However, lithium-ion battery is very susceptible to battery explosions, and hence, it costs lives and properties. So in order to solve this one, aside from the safety issues of the lithium-ion batteries, lithium is also considered as the gold of the next century due to its limited abundance, increasing cost, and the wide range of applications. And hence, researchers are doing a lot of works to find an alternative battery to replace that of the lithium-ion battery. So one of which is that of the sodium-ion battery. Sodium-ion battery is a potential candidate to replace or um, to to replace that of the lithium-ion battery due to its natural abundance and low cost. So in a simple battery setup, instead of using lithium-ion, we will be using sodium-ion as well as sodium-based material as cathode material. However, similar to that of the lithium metal, sodium is very reactive with that of the air and moisture and thus resulted to an ignition, as well as it's very reactive with that of water. And hence, it needs vacuum technology to manufacture. In addition, sodium-ion battery also needs a host or matrix to hold the sodium ion, as well as it uses an organic or ionic liquid as electrolyte. So in order to solve the safety issues, researchers are looking for an alternative for that of the lithium-ion battery, one of which is that of the zinc-ion battery. Zinc is known as to be naturally abundant and low cost. In addition, zinc is stable in air and moisture that prevents the ignition or the combustion of batteries. So in addition, since zinc is not sensitive to air and moisture, it can be manufactured at room condition without the use of any sophisticated equipment. So to summarize or to compare the different types of battery that I'm currently considering, the sodium-ion battery have lower volumetric capacity and gravimetric capacity compared to that of the lithium-ion battery. Sodium-ion battery have higher abundance and lower cost compared to that of the lithium-ion battery, which make it a possible alternative to lithium-ion battery. On the other hand, lithium-ion battery have lower gravimetric capacity, but comparing the volumetric capacity, as you can see, it is twice as that of the lithium-ion battery. In addition, zinc is naturally abundant and it has low cost, and since zinc is environmentally stable, it can be manufactured at room condition. In addition, zinc-ion battery can also utilizes non-toxic solvent such as that of the water. So the overall advantages of this zinc-ion battery makes a possible alternative to zinc-ion battery. So the objective of the study is to utilize 1D vanadium oxide compound as ion storage for lithium-ion battery, sodium-ion battery, and zinc-ion battery. So vanadium oxide batteries, vanadium oxides are widely used ion storage due to the multivalent state of vanadium, as well as it provides a conductive backbone to the material. Vanadium can also form an amorphous oxide matrix to buffer the large volumetric change. So there are a lot of morphologies that are being considered for different materials to enhance electrochemical properties, one of which is the one-dimensional nanomaterial. So for one-dimensional geometry, it allows an efficient electron transport compared to that of the small spherical nanoparticles. As you can see, for small spherical nanoparticles, there's a random walk of electrons compared to that of the one-dimensional, wherein there's a one-direction flow of electrons. In addition, smaller diameter can facilitate strain relaxation during insertion and removal of ions. And lastly, the nano-size dimension and high surface area to volume ratio allows the short lithium-ion diffusion length and large lithium-ion flux. So for the first part, we investigate the porous ZnV204 nanowire as anode material for lithium-ion battery. So for this step, we fabricate porous ZnV204 via two-step fabrication. Initially, there's already an existing study which forms the one-dimensional ZnV206. However, based on their study, which uses hydrothermal process, it took a lot of time, which is approximately 7 days. So for our study, we um, reduce the reaction time to 24 hours by the addition of DTAP. So after we produce the ZNB206, we calcine the sample under 3% H2N2 to produce the one-dimensional ZNB204. So based on our result, based from the XRD pattern, as you can see, after the hydrothermal synthesis, the produced particle is ZNB206 with prominent peak at around 29.356 degrees, which means that the surface of the particle is the dominant plane is 200 for the surface of the nanoparticles. So based on the SEM image, after hydrothermal process, the formed particles is that of the one-dimensional nanomaterial. So after calcination, as you can see, this ZNB206 was converted to ZNB204. So this ZNB204 was able to maintain the wire-like morphology of that of the ZNB206. So to check the purity of the sample, we examined our sample under XPS and found that the oxidation state of zinc is that of the +2, while that of the vanadium is +3. So based on our result, there's a 4.53% +5 vanadium. So this can be due to the surface oxidation as well as the incomplete reduction of ZNB206 during calcination. So to further analyze our sample, we examine our sample under TEM. So the wire-like morphology that we observe has a porous structure. As you can see, there's a lot of pores. Though it has a porous structure, based on the SAED analysis, it is still single crystal that grow along the 002 direction with 220 facets. So this analysis agrees well with that of the STEM images. So based on the STEM images, the surface of the nanomaterial have a 220 plane, which indicated by the distance between the lattice fringes, which is approximately 2.97 amperes, and that of the hexagonal pattern on both HADEF and the ABF images. So the hexagonal structure of both HADEF and ABF images correspond to the 220 plane of the ZNB204. So this hexagonal pattern is composed of 10 atoms, 6 of which is that of the zinc, and the 4 less prominent peaks or less intense atoms is that of the vanadium, while the central atom is that of the vanadium atom. However, oxygen cannot easily be seen or cannot easily be detected using the HADEF, and thus we use ABF to determine the exact location of the oxygen atoms. So based on our ABF, we have a total of 6 oxygen atoms surrounding that of the central vanadium atom. So these porous ZNB204 were utilized as anode material for lithium-ion battery. So based on our result, the formation, there is a sudden decrease of on the capacity from the first to the second cycle. This can be attributed to the formation of the SEI layer. On the other hand, from the second to the 35th cycle, there is a gradual increase in the capacity, which can be attributed to the phase transformation. And lastly, above 35th cycle, we have a very stable cycle until the 100 cycle, which makes it efficient anode material for lithium-ion battery. So the advantages of this porous one-dimensional nanostructure is that of the 1D flow efflection, as well as it can facilitate strain relaxation. In addition to the advantages of one-dimensional nanomaterial, this structure have a porous structure which can shorten the lithium diffusion path and improve the surface area, as well as allow higher lithium-ion flux that is favorable for high current density application. So these porous ZnB204 were also analyzed as anode material for sodium-ion battery. So based on our result, it can be seen from the SANES that the oxidation state of zinc is still +2, similar to that of the XPS result, as well as that of the vanadium, which has a +3 and +5. The +3 is still due to the surface oxidation. And for the PET analysis, it can be seen that the porous ZnB204 follows the type 2 isotherm, which means that has restricted monolayer multi-layer adsorption. On the other hand, this porous ZnB2O4 has a hysteresis loop of type H4, which means that it has a mesoporous structure, which agrees well with that of the TEM image. In addition, on this experiment, we considered three types of temperature, calcination temperature, that is 500, 600, and 700 degrees Celsius. As you can see, based on the BET analysis, as the calcination temperature increases, the surface area as well as the pore size also increases. So these three samples were examined under the as anode material for sodium-ion battery. So based on our result, we found that as the calcination temperature increases, the capacity also increases. This can be attributed to the higher surface area of the produced nanoparticle. So in comparison with the other study, this work, or the porous ZnB2O4, has higher capacity compared to that of the other material, which makes it a good anode material for sodium-ion battery. So for the zinc-ion battery, instead of using ZnB2O4, we utilize V2O5 nanofibers. So on this synthesis, we utilize a binder-free V2O5 wherein we directly grow the V2O5 nanofibers on carbon cloth without the use of any binders. So for this synthesis, based on our result, we have a fibrous V2O5, or have a fibrous V2O5 that grows along the 001 direction with 010 and 100 plane at its facets. This can be explained by the difference in the surface energy. As you can see, the 100 as well as the 010 plane have lower surface energies compared to that of the 001 plane. And hence, the growth direction will be towards the 001 plane. So to examine the purity of our sample, we examine our V2O5 under XRD and XPS. So based on our result, the produced sample is a pure V2O5 compound without any impurities. So we examined this sample under galvanostatic charge and discharge cycle and found that V2O5 nanofiber is a stable cathode material for aqueous zinc-ion battery. So as you can see, it has a very stable capacity until 200 cycles, as well as it has a good capacity retention even after the 3 repetitions. We also observed that despite the high solubility of V2O5 in water, V2O5 did not significantly dissolve during the charge and discharge cycles. So to summarize, vanadium oxide-based material is a good charge storage material for lithium-ion battery, sodium-ion battery, and zinc-ion battery due to their multivalent states. 1D nanomaterial facilitates strain relaxation that buffers the large volumetric change upon successive charge-discharge cycles, and porous structure yields higher surface area and thus results to higher capacity. So there's already a direct-grown binder-free nanowires is an economical, eco-friendly, and excellent electrode material for rechargeable battery. And most importantly, aqueous zinc-ion battery is a potential battery alternative to the commonly used lithium-ion battery, especially for addressing the sustainability, cost, and the safety concerns of the rechargeable batteries. So if given a chance to obtain a grant, I will continue with this research, future researches, with the following. So first, I will examine the biochar-based cathode material for zinc-ion battery using an indigenous material. Second, I will examine a flexible zinc-ion battery for flexible electronic application. And lastly, I will examine a high operating voltage zinc-ion battery for high energy density application. And that ends my presentation. Thank you for listening.
Thank you, Dr. Corpus. So our fourth presenter is Dr. Wilfred John F. Santañez, and he is presently an assistant professor at the Marine Science Institute of the University of the Philippines Diliman. He completed his doctor degree in natural history sciences in 2018 at the Hokkaido University, Japan, same as Dr. Corpus. Let's all welcome on screen Dr. Wilfred John E. Santanyes as he presents his paper on Untangling Nets, Elucidating the Diversity and Phylogeny of the Clatrate Brown Algal Genus Hydroclatrus with the description of a new genus, Tronoella, did I say it right? Cytospinaceae Phaeophaeaceae. Okay, Dr. Santanyes, please.
Right. Good morning, everyone. So I'm Wilfred John Santianes of the UP Marine Science Institute at the in Diliman, and I'll be sharing today my paper entitled Untangling Nets, Elucidating the Diversity and Phylogeny of the Clastrate Brown Algal Genus Hydroclatrus with a description of a new genus Tronoella Scytosyphinaceae phaeophyceae. First, what is Hydroclatrus? Hydroclatrus are brown seaweeds that are easily recognizable even in the field because of their net-like appearance, which is a result of the formation of numerous holes in their thallus. So a thallus or a thalli is what you call the body of your seaweeds. Aside from their ecological importance, particularly as primary producers, hydroclatrus are economically important. They are consumed as food, particularly by populations in northern Philippines. They are also sources of high-value natural products such as alginates, which is the main component, for example, of Gaviscon, which can be used to treat heartburn. They can be used also as fertilizers, feed and fodder, and has potential for biofuel feedstock for biogas production. So, like any marine algae or seaweeds, they also possess bioactive compounds and for various applications, including the medical, pharmaceutical, and nutraceutical industries. Globally, there are 4 species that are known under the genus Hydroclatrus, and the most widely distributed is the generic type Hydroclatrus, followed by the Indo-Pacific species Hydroclatrus tenuis, and then by the Central Pacific Hydroclatrus tumulis, and relatively limited species with the distribution in Australia and New Zealand, Hydroclatrus stephanosaurus. Now, there is an existing problem as to how to correctly identify the different species of Hydroclatrus in a consistent manner. With respect to usually when you find Hydroclatrus that has a thick and robust thalli in the field, you would attribute that to Hydroclatrus catratus. But when you have a thin and fibrous thalli, you would attribute that to Hydroclatrus tenuis. However, worldwide reports of these 2 species show that they have a wide overlap of morphological characteristics, such that Kraft and Abbott in 2003 advocated the use of DNA fingerprinting to finally settle the distinction between these 2 species. Later, in 2009, Kraft also noted on the wide overlap of the characteristics of Hydroclatrus stephanosaurus with those of Hydroclatrus catratus, and suggested the reassessment of the taxonomic status of Hydroclatrus stephanosaurus. So now we have a problem in identifying the 3 different species out of the 4 Hydroclatrus worldwide. Now, why is it important for us to be able to consistently identify our seaweeds? This has practical application, particularly in the seaweed industry. Names of seaweeds would introduce or enhance the value of a seaweed crop, and they would also announce the kind and quality of product that a seaweed produce. So in the farm seaweed, Capophycus and Eucuma, both of them possess carrageenan or produce carrageenan, but of different types. So carrageenan is a very important gelling agent that is widely used in the food industry. So if you brushed your teeth earlier today, I hope you do brush your teeth at least twice a day. Um, the gel component in your toothpaste is actually primarily made of carrageenan. So if I am a seaweed farmer, if you can see the figure, Kappa ficus commands a higher price as compared to those of Eucuma. So knowing that I am farming Kappa ficus would have a greater incentive for me because the return of investment for my efforts in farming this seaweed is higher as compared to when I will use or farm Eucuma. So if I am a seaweed processor, particularly, I would be very particular as to whether I am processing for Kappa ficus or Eucuma because both of them, while they produce carrageenan, individually they produce different types of carrageenan. So Kappa ficus produce a hard gel and Eucuma produce a soft gel. And mixing them both would be problematic for me as I won't get the desired gelling properties of that I would want to produce. So that is the practical application of knowing what the name of your seaweed is. Now, back to hydroclasrus, we attempted to settle the distinction between the different Hydrocratus species by using a total of 117 Hydrocratus samples collected from the known distribution range of Hydroclasrus. So we analyzed them based on morphoanatomical and molecular data. So we extracted first DNA sequences and conducted phylogenetic analysis. And from the phylogenetic analysis, we delineated species lineages and then conducted morphological observations on representatives of those species lineages. All right, so based on our COX-3 sequence data, we were able to delineate 5 species lineages that we attribute to the genus Hydroclasrus. And we found that 2 of these lineages were found in localized areas, while others are widely distributed. When we look at the phylogenetic relationships or the relationships of these different lineages in relation to other members of the family Scytosiphonaceae, we found that these species that we attribute to Hydroclasrus does not form a single cluster. So ideally, we would want that all our species would cluster into one group so that it will have a natural classification. So in this case, we noted that Hydroclasrus is not a monophyletic group, but nonetheless, we were able to morphologically identify, we noted that these different lineages are actually morphologically distinct, and we were able to assign names to 3 of these lineages. So in order, the criteria for us to assign names is that first, it should have, the representative species would have a very strong resemblance to the original description of the species. And the other one is that we should have a sample that was with genetic data that was collected near or within that locality from where the original species was described. So for these lineages, we were able to confidently meet those criteria, so we have assigned them with these names. However, for the 2 other lineages, we were not able to assign name, and we describe them here as new taxa. So for the first one, we name it Hydroclasrus minutus because it is the thinnest and the smallest among the other Hydroclasrus. It's only less than 130 microns thick. And one of the distinguishing characters is that it's pleurangia. So the pleurangia is your reproductive structure for your brown seaweed. So they are arranged in circular sori surrounding hairs. And then the medullary cells, so this white or bluish, light bluish cells are thick-walled. So typically, your Hydroclasrus would have a thin-walled cell. So this species is also found in southern Japan and in Vietnam. So for the other seaweed lineage, it appears that it is phylogenetically distinct from other Hydroclasrus species, and it seemed that it has diverged much earlier than other members of Hydroclasrus, showing indications that it is possibly a new taxon rather. So when you look at the gross morphology of this species, it closely resembles other Hydroclasrus, but when you look more closely, it actually has these numerous protrusions. Which is not found in any of the Hydroclasrus species. Now, when you make a cross section on that protrusion, it is actually hollow in the center. And when you also observe that it is cleaved along its length, and that the cleaved portions becomes inroll later. Among the distinguishing character of this species is that the plurangia is actually in clumps, so they are firmly coherent and massive, and contrast that to those of Hydroclathrus, which are typically arranged like a baseball bat. Okay, so based on the unique phylogenetic position and the unique morphology of this species lineage, we actually named. We decided that it is a new genus, and we named it Tronowella. So Tronowella is after my mentor at the UP Marine Science Institute, Dr. Gavino C. Trono Jr., who is also one of our national scientists here at the NAST. We named it after him to honor his contributions to the understanding of the diversity and ecology of the seaweeds in Central and Western Pacific. So we named the species as Tronowella ryukyuana. So ryukyuana is after the kingdom of Ryukyu, from which Okinawa belonged previously before it was annexed to Japan. All right, so with respect to the global distribution of Hydroclathrus, we've shown that Hydroclathrus cathratus is indeed a widely distributed species, but it has a disjunct distribution. We attribute this disjunct distribution on sampling artifact. Now, for Hydroclathrus stephanosaurus, we noted that it is more widespread than previously known. So before, it was only just found in Australia and New Zealand. And in this study, we actually reported it to be widely distributed in Northern Pacific. And it's also the first time that it was recorded in Northern Atlantic. Right? So for H. tenuis, it also seemed to favor the warmer waters as compared to the other two species. Now, in summary, we delineated here all the known Hydroclathrus species from the three ocean basins using an integrative approach. And we also provided insights on the geographic distribution ranges of each species using globally sampled data. For this research, we also described two new taxa that are new to science. So one is under the genus Hydroclathrus, which we call Hydroclathrus minutus. And the other one is a new genus, which we named after National Scientist Gavino Trono Jr., which we named Tronowella, and the species is Tronowella ryukyuana. Among the breakthroughs of this research is that it is the first seaweed genus that is named after a Filipino scientist. So this is the first time that has been made, and it in recognition of the role and the contribution of National Scientist Trono to the global seaweed research and development. And we also provided here a framework for taxonomic and classification revisions within the family Scytosiphonaceae, which later would translate into the description of three new taxa within the family. So one, we added another species under Hydroclathrus from Easter Island in Chile, and we call it Hydrocrathrus rapanui. And we also describe additional two new genera. So one is the Pseudonoospora, which is found here in the Philippines, and the other one is Dactylosiphon, which is a warm temperate genus. Additionally, we also generated foundational knowledge that can help us in ensuring the correct identities of seaweeds that we can use for seaweed resources development, particularly Hydroclathrus and other members of this brown seaweed family. So what can we glean from this research? Among the major insights that we provide here is that global marine biodiversity remains poorly known. And as we demonstrated here by describing and discovering new seaweed species, it is apparent that many marine species are still hidden in plain sight. And the apparent lack of our knowledge and understanding of marine biodiversity resources puts us in a disadvantaged position, primarily because it limits our focus to few species that we can actually tap, for example, for mariculture and development. And then it also hinders our capacity to actually tap the potentials of this resource for the benefit of the society in general. And lastly, it restricts our capacity in determining local and global marine conservation priorities. And note that these priority areas are actually selected and anchored on our knowledge on what we have, where we have them, and when we have them. So as it's always been said, we only protect what we love, and we only love what we know. So that brings me to seaweed biodiversity and systematics research in the Philippines. And it was really encouraged that we actually put more effort and boost our support in this field, primarily because there are so many seaweed resources that are yet to be tapped that we can use for sustainable development in the Philippines. And with that, I'd like to thank and acknowledge my funding. So this study is primarily funded by the Japanese government through the Mumba Kagakushu Scholarship Grant. I also wish to thank my professor, Kazuhiro Kogame, and the members of the Algal and Protist Systematics Laboratory of Hokkaido University in Sapporo, Japan. And the global sampling was made possible by my collaborators from around the world, and I cannot name them all here. So, and also I would wish to thank the members of the Marine Phycology Laboratory, my research staff and students for their encouragement and support, and the Marine Science Institute for the trust and the opportunity to actually raise leaders in seaweeds research, UP Diliman for UP System, actually for the UP Balik PhD program, and for the funding, and DOST Picard for being a host for my long-term engagement as a DOST Balik scientist. I also wish to thank my family, my mother and father, and my friends in Diliman and abroad. Thank you.
Okay, so we're now down to our free presenter, but definitely not the list, no? So Dr. Somesh Artus and Maini is also a homegrown talent, obtaining his Bachelor of Science and Master of Science in Biology from the Ateneo de Manila University, and currently taking his PhD in Biology at the same institution. His research entry contributes to the field of bioremediation, the use of living organisms and their products to treat and manage polluted environments with the goal of rehabilitation and eventually for restoration purposes. A round of virtual applause for Dr. Somesh Artus and Maini as he presents his study on lead-2 tolerance and uptake capacities of fungi isolated from a polluted tributary in the Philippines.
Hello, good morning everyone, to the panel members and to my co-presenters and to everyone who's watching. Actually, hindi pa po ako doctor. I'm still a PhD student. I'm still studying. And I come from the Ateneo de Manila University, Department of Biology, and I'm very happy and thankful to be able to present this paper entitled Lead Tolerance and Uptake Capacity of GI Isolated from a Polluted Tributary in the Philippines. So in 2007, the World Health Organization released a comprehensive paper that details the impacts of various anthropogenically released metals. Among these metals, you have arsenic, cadmium, mercury, copper, and lead, which were shown to be detrimental to human health because they pollute our water systems. Among all of these heavy metals, lead is considered to be a causative agent of disease and disability. This is because lead has no known physiological use and is toxic to all life forms, even in very, very tiny quantities. Given this, it's a bigger problem because we actually experience 1,000 times more lead burden today compared to the very first modern humans, which of course translates to a higher incidence of disease. In fact, just last 2015, the World Health Organization reported 495,000 cases of death from lead-related illnesses. And these are only the reported cases, not even all of the cases. So if we believe that the problem with lead is a health concern only for developing countries that have poor heavy metal mitigation strategies, we're actually very, very sorely mistaken. Just 2 years ago, thousands of different areas in the United States were afflicted with lead poisoning because of contaminated water reaching various households. In the Philippines, over the last 2 decades, there have been several rural and urban areas that have recorded elevated levels of lead. A good example of this was a 2015 paper published about a village in Marilao where 9 out of 10 children were confirmed to have elevated lead exposure, which was thought to be because of flooding that affected their protected areas. Very recently, just last October 2018, an independent study by the Coalition found that various playgrounds all over the country, imagine kids playing here, had elevated levels of lead. Because of this, we can see that the problem of lead is something that's pervasive. It's been there for a long time, and yet the existing strategies in order for its mitigation remain inadequate because these are costly, time-consuming, and ineffective. This actually opens the door for the use of bioremediation, technical and this one as an alternative. And opens the door for the use of bioremediation as an alternative strategy for the management of heavy metal waste. You have to be examined their lead. And among all of them, fungi actually have generated their ability to be able to suck up higher metal concentration in a variety of different organisms. Now, how can fungi do all of these things? Fungi are able to do bioremediation mechanisms because they have natural tolerance mechanisms and acquired resistance characteristics that they can use for them to tolerate higher heavy metal concentrations. Innate properties such as the presence of metal affinity binding sites on the fungal cell wall. Metal affinity binding sites on the fungal cell wall that confer a natural tolerance that is not necessarily because the fungus is exposed to a heavy metal. You can also have various reactive mechanisms such as the production of ligands that confer increased resistance upon exposure to heavy metal concentrations. Together, these tolerance and resistance mechanisms contribute to the survival, growth, and reproduction of fungi in elevated heavy metal concentrations, which of course can be exploited for use in bioremediation. Now, given all of this, the key goal of this paper was to isolate local lead-tolerant fungi and measure their potential for the uptake of lead. Specifically, this paper aimed to provide the identity of these local isolates, to be able to screen them for resistance against increased metal concentrations in solid media, and to be able to quantify their lead uptake abilities in liquid medium as well. The methodology that was utilized by this paper in order to achieve all of these goals can be broken down into 4 major segments. The first segment would be a preliminary screening that involved the serial dilution of environmental samples in order to be able to yield pure fungal isolates that could survive at 1,000 microgram per ml of lead. Once these pure fungal isolates were already culturable, what we did was we subjected them to fungal identification, followed by a tolerance and resistance test, and then a lead uptake test. Now, the key results of this paper mirror these 3 particular test parameters. The fungal identification was carried out using PCR targeting the internal transcribed spacer element sequences of each of the particular isolates. And the results of the sequencing were corroborated and supported using the examination of various morphological structures. For the tolerance and resistance test, the mycelial diameter and growth of each of the fungi on metal-supplemented medium and non-metal supplemented medium was examined, and the ratio yields a value called the tolerance index, which is supposed to be reflective of the relative growth of each of the isolates on solid media. Finally, we performed lead uptake tests that could be broken down into a removal efficiency test and a total uptake test. The removal efficiency test measures the percentage of lead that is removed from a solution with a predetermined concentration of the heavy metal. The total uptake test, on the other hand, quantifies the amount of lead per gram of fungal biomass. Together, both removal efficiency and total uptake are going to be heavily dependent on a technique called inductively coupled plasma atomic emission spectroscopy in order to be able to measure the residual ion concentrations in the solution after a 15-day incubation period. So, jumping to the results of this work, we were able to isolate 4 particular fungal species 2 from the genus Penicillium, and 2 from the genus Thalaromyces. The taxonomic identities based on sequence similarities found on the NCBI database is here. You can see it in table number 1. Now, going through the tolerance index test, the second major parameter tested here, what we generally can see is that all isolates are capable of growth at 500 to 2,000 microgram per ml of lead. This is already astounding because most measured tolerances for heavy metals usually range at around 10 to 100 or maybe even up to 500, but never going past around 1,000 microgram per ml. So the very high tolerance indexes that are shown by the different species, as can be seen by values of TI greater than 0.5 in all of the curves, are a testament to innate and adaptive strategies that these different fungi use for tolerance. Of course, different fungi are going to have different means by which they perform all of these particular mechanisms, and that is why you will be able to see variations in all of the curves presented in this particular figure. However, as we move to 3,000 microgram per ml, we see that growth is depressed for 2 isolates labeled B and D, and 2 other isolates labeled A and C are completely inhibited. This is not something that is not expected because the susceptibility of various fungi to increased heavy metal concentrations has been documented in various literature. However, we also have to remember that we're testing at 3,000 microgram per ml. So that's very, very high, and is something that is not seldomly reached naturally in any particular environment. Finally, we can see that for both Penicillium canescens and Penicillium simplicissimum, there is going to be elevated tolerance index values at 500 microgram per ml of lead, which is actually an indication of enhanced growth. So this is not something that's also common to a wide variety of fungi, but it has been recorded in some Penicillium species. This may mean that the progenitor fungi that released these spores, which we were able to collect, were actually exposed to the heavy metal lead for an increased amount of time and thus produced spores that were capable of tolerating it at this particular concentration. Now, moving to the removal efficiency, we can see that the removal efficiency is going to be either increasing or consistent for most of the different species up until 2,000 microgram per ml, and for Thalaromyces macrosporus, that's letter C, up until 3,000 microgram per ml. This percentage R consistency denotes the uptake stability of each of the different isolates up until these particular concentrations, indicating that they're actually quite good for usage in terms of accumulating the heavy metal lead in liquid medium. Among all of them, Thalaromyces macrosporus is going to be the best performing with an R value that does not change statistically even if you increase the heavy metal concentration. Its R percentage range is going to be around 97.9 to 99.5% across all of the test concentrations. Now, the last and most important portion of the paper would be the discussion on the uptake capacity, or what we call the Q value of all of the different species. So the first thing that we notice here is that Penicillium canescens and Thalaromyces macrosporus were originally unable to grow on solid media, but were able to grow here in liquid medium supplemented with the same concentration of lead. This is probably due to better air perfusion and mixing that allows greater nutrient accessibility, which makes the fungi not susceptible anymore to this particular concentration. So the most important result of the entire paper is that we see that total uptake is going to be increasing as you increase heavy metal concentrations from 500 microgram per ml of lead to 2,000 microgram per ml for all of the different species, but up to 3,000 microgram per ml of Thalaromyces macrosporus. So what does this mean? The only way by which you can have increasing uptake values like this and you have consistent removal efficiency values is if the fungus does not increase its mass, does not have cellular proliferation, and yet continues to uptake the heavy metal to be able to remove the 90+ percentage that was shown earlier. This means that whatever the fungus is doing, it's not only a biosorptive mechanism, but it can actually also be a bioaccumulation mechanism that is activated in the presence of the increased heavy metal concentration. Finally, what we can say is that Thalaromyces isolates outperform the Penicillium isolates, and Thalaromyces macrosporus can uptake 601.0 milligrams of lead per gram of fungal biomass. This is an extremely high amount, as you can imagine, because if you get 1 gram of the biomass, more than half of it is actually going to be elemental lead. These are new records for all of the species, and these are the first records for both Thalaromyces isolates as well. So that's one of the major contributions of this particular paper. So in order to summarize the content of my presentation and my paper, let me just walk through again what we've been discussing. So we saw that lead pollution is a persistent ecological and health problem. We saw that fungi can be utilized as alternatives to remediate heavy metals. We were able to isolate local fungi that have natural tolerance capacities, and we were able to show that these isolates possess removal and uptake capacities as well, which means that they can actually be further developed for remediation purposes. But what kind of application can you expect with a basic study like this? So usually we would like to see something industrial. We would like to find something that can be used in industry. But the thing is, to be honest, the use of living cells for bioremediation on an industrial scale is going to be very, very difficult. Well, because they're living cells. You're not going to be able to produce enough biomass in order to treat something like a river, for example. This is why we recommend that we shift our perspective from total uptake to purely biosorption. And if we are able to get dead fungal biosorbent and immobilize it in a matrix, we can create some sort of material that eliminates problems with living cells such as cell fragmentation and mechanical weakness. In fact, we have a second published paper just recently that did this particular thing, where we got Thalaromyces macrosporus and embedded it in a matrix made out of wood, and we were able to remove 99.5% of all lead in a liquid solution in 30 minutes. So that can be something that can be developed for industrial application. Finally, if there are other researchers who are interested in doing something like this, then one avenue that can be further developed would be trying to understand the how. How can the fungi do something like this? How can they tolerate increased heavy metals? Or how do they adapt? Which means you have to take a look at genetic mechanisms. And you have to understand these genetic mechanisms as a tool in order for us to benefit from the resistance that these fungi show. Fungi are also eukaryotic organisms, which are very similar to us, which means that the pathways of resistance that they have might be analogous or homologous to some of the pathways that we possess, but currently underutilized. With that, I end my presentation, and these are my references, and thank you very much for your time. I would like to acknowledge the University Research Council of Ateneo for giving us the money to do this, and the Departments of Biology and Environmental Science for the use of their facilities. I'd also like to take this time to thank all of the undergraduate students who are co-authors of this paper, because it's actually quite tedious to keep on culturing and testing. And they really spent a lot of time, even weekends, in order for us to be able to come up with a publishable paper. So again, thank you very much, everyone, and I'm very happy to conclude my presentation.
Okay, thank you everybody. So let us all give a round of applause to all our finalists. Okay, we shall now proceed with the open forum for the question and answer portion. Our Board of Judges, you may now proceed. So at this point, may I call on the 5 finalists to occupy the 5 seats now being arranged on the stage. Okay, to our Board of Judges, you may now ask for your questions. Sir, ma'am, you have questions for our finalists?
I have.
Dr. Padilla?
Yes. Well, thank you. So allow me to ask the first question to the first presenter. I actually have questions for the first and the second. So for the first presenter, first, congratulations to all of the speakers. Okay, so congratulations to all. It's really nice to see a very young group, presenting this kind of work to us. So I appreciated really the work on the coconut because I've been monitoring the coconut genome. Okay, so I have been actually been following up the work on the coconut genome. So I really appreciated your update on this work. So I just want to check are you part of the coconut genome project of the Philippine Genome Center? That's one. And second is you mentioned a lot of findings out of your project. Anywhere from the resistance genes to the pest and then to the variants. So if you were to choose maybe the top 3 that you'd like to recommend to government, which one will it be? And what is the extent of your work right now? How far have you gone with getting your work to the stage of being tested by the farmers? Like, have they been, how far have you gone? Because this is just the basic work. This has to go through so many steps yet. So can I ask the first speaker, Darlan Lantican?
The first question is you're asking me if I'm part of the Philippine Genome Center's. Hello po?
Yes. Is your work part of the Philippine Genome Project? Because
Of course.
Yes, ma'am.
okay.
Obviously.
Okay. All right. Okay. So may I know now, among all of the genes that you actually mentioned, which ones would you recommend to government to prioritize so that we can actually move this to the market?
Actually, the SNP variants that was developed, the SNP variants, ma'am.
Okay. And for which one?
Variants is actually the unique output of this project, wherein we were identified around 50,000 variants that can be targeted for genotyping by sequencing approach. And this will open a lot of opportunities in terms of genetic improvement, like the application of advanced plant breeding methods, for example, the genome or genomic selection that could facilitate the development of coconut varieties at a minimum amount of time. As compared, if we go to the conventional method of breeding coconut, it needs a whole lifetime just to develop one variety. But here with genomic selection, we can actually lessen it to about 20% if we have a good training set of utilize the information of the genes.
Yes, Darlan, but which of this information has moved to the next step? Because this is which one?
Ma'am, maybe the oil genes, the most economically important part, the oil genes, because we found some SNP on the LPAT. It's a major pathway for the development of medium chain fatty acid, particularly the lauric acid in coconut. So we can further improve now the oil yield of coconut using the particular SNP. And at the same time, we can have we call it the background markers in order to precisely integrate that particular gene on our elite varieties.
So how soon will we see this? Can you give us a timeline?
Sorry, ma'am.
How soon? How soon will we see this in being used by the farmers?
The problem will be funding, of course, because genome or GBS or genotyping by sequencing approach would take a lot of money, of course. But if we can now start again the sequencing of other varieties of coconut, not just the Katigan dwarf, then we can have a lot of opportunities to further improve other varieties, not just the dwarf, or at least the hybrids or the other calls that we have in the coconut class.
Okay, because it's a waste if this thing will not go to the next level. So maybe after this conference, maybe we can have another discussion. So I'd like to speak to you after the meeting. Okay. I just have one other question before I move on to my other meeting, actually. The question is really for the second speaker on cancer. I just want to know how far you've gone, because definitely the marker for cancer, your work on the magnetic, I mean, it's something we need. We need diagnostics, but how far are we to using this probably in the clinics on the next step? How far are you? I just want to know the application part.
Hello, ma'am. So good morning. Thank you for your question. So this is actually part of the project that I did in United Kingdom for my PhD work. And my project already is we're done with the proof of concept, okay?
Okay.
So basically, we are now in the application step. However, it is my group that in UK who is doing this already. So it's a research culture in the lab actually to obviously like continue this research work, something like that. So if this is, I think before I left, because I also was involved already in the application step, they are now using real samples of patients
Okay.
from the hospitals in the National Institutes of Health in the United Kingdom. So they were collecting. I was actually involved in, let's say, DNA extraction before I left UK. So in terms of, let's say, how far we are, I think I will give you, let's say, a year let's say less than a year, let's say 6 months, for example, to fully implement this in real samples, real world samples.
Okay, thank you.
Thank you.
I'm Dr. Torres. My field is computer science, and I'd like to ask a question to the fungi thing. My question has to do with how do you store this knowledge that you have gathered so that further studies by other people might be enhanced, no? Because of not having to go through the process that you went through. Is it only the report that will be seen by further researchers, or there is a bunch of data and insights about that data that have been stored so that it will speed up diba, knowing what has been done and move on to new things? That's my first question.
So when we submitted the paper, the reviewers didn't really ask for all of the raw data anymore, because actually the raw data is quite voluminous given the number of replicates and the number of repeats and the number of trials as well. So we haven't really uploaded that because we never really thought about what you just said, that there might be someone who would be interested to look at the measurements over again. But I know of certain ways by which data like that can be uploaded. So maybe that's something that we can do after this presentation.
Okay, well, I'm not interested for myself, right? I'm interested in how you do that, and perhaps this is also related to the other presentations, where when there are lots and lots of data, I think an important step in research has to do with storing this data properly so that others can benefit from not having to gather all this data again. And in other words, it's a stepping stone to being able to move on. And that, I think that technique is already more and more available today. And maybe even you, when you do further research, you can benefit from having a readily accessible data, automated, that can be accessed maybe using things like machine learning and artificial intelligence. These are techniques that are difficult to do on a manual basis because of the volume of data. And of course, there will be continuing work in terms of gathering data, and maybe it would also prevent people from having to do the same thing already done. So that's what's behind my question. I think that question also applies to the cancer study.
Sorry, sir.
Okay. The other question I have relates to the coconut study, on the dwarf coconuts. At the beginning of your presentation, you mentioned that the productivity in other countries that you mentioned is much, much higher than the productivity in our country. So my question is, will your study enable Filipino coconut farmers to catch up?
Actually, sir, the resources that can be developed or that was being available using this study can be used for the hybrid breeding programs because mostly our coconut plantation in the Philippines are senile or not on the optimum yield anymore. So we need to perform replanting. And during planting, maybe we can use the hybrid or the hybrid varieties that is actually has a higher variety, has a higher yield. So we at the Philippine Engineering Center through the funding of the USSR funded this research actually to further increase our yield per tree to about 150 nuts per year. So that's actually our goal. Use
which is
hybrid
still
technology.
behind the goal that other countries you've mentioned kasi nasa 300 na sila, di ba?
Yes, sir. Opo. Pero
And,
there's
may I add, these people will also make use of your results,
yes,
perhaps.
sir. The
And so
resources.
we will be trailing them all the time.
Yes, sir. Actually, we are in collaboration with the Philippine Coconut Authority through Dr. Manohar and through Erling Manohar, the one of the regional coordinator or the deputy director for the Philippine Coconut Authority. So we are accepting genotyping services in order to perform hybrid testing on our coconuts in the Philippines during their replanting programs. So we have actually, supposedly, we will start the hybridity testing, um, but there's a COVID pandemic, so we didn't perform it. But we are positive enough that we can perform the hybridity testing for the replanting programs,
Okay.
possibly along the Calabarzon area first before the whole Philippines.
Thank you very much. I have just one last question for Dr. Corpus on the batteries, storage batteries. No, you are in an important field because humanity really needs power, and much of this power, especially the naturally sourced power like sunlight and wind and so on, they are seasonal, and therefore storage is an important factor. So my question to you is, how far are we, or what are the prospects of improving the storage of energy in the batteries because of the result, as a result of your research?
Currently po, sir, I'm doing the zinc ion battery. So for the zinc ion battery, it's still a new type of battery wherein we're still doing the coin type cell. So for the large application, it's still under my future researches po.
Okay. So, uh, can we expect improvement in the large storage, the large capacity storage, as a result of what you have done?
Currently po, based on my current results, not yet po, but maybe after 2 years, we can have that one po.
Okay, thank you.
Thank you.
Okay, National Scientist Raul Fabela would like to ask question.
Now, this is returning to the renewable batteries issue. There may be about maybe hundreds of teams working on the issue at the moment, and excitement always builds when the results touches on either of these three properties. One is density, the other is cycling properties, and of course the third is the cost. Which of these properties you think will be most impacted by your research on zinc oxide? I understand the zinc oxide could improve all types of battery chemistries, whether they are lithium ion or some others, right? So which of the three important properties would be impacted by your results on zinc ion batteries?
For the zinc ion battery, thank you for your question, sir. So for the zinc ion battery, it will cover all the properties since for the volumetric capacity, it has twice that of the lithium-ion battery. So it's much better in terms of volumetric capacity. For
What is
the
volumetric? Is that also density? Density,
volumetric
that also
specific
density?
capacity, volumetric capacity po. For the volumetric
That,
capacity, it is more than twice that of the lithium-ion battery. So it's much better in terms of volumetric capacity. And then it is stable it is abundant, and then it has low cost, as well as it is very stable in air and water, so it can be manufactured at room condition. So it has a great potential as an alternative for for lithium-ion battery po.
this is intended to be the anode of the lithium-ion batteries.
Come again po? No, it's not the anode of the lithium-ion battery.
The
It's a
what?
different type of battery po. So different, same concept as that of the lithium-ion battery, but instead of using lithium, I'm going to use zinc ion and then zinc anode. So without
Zinc
any
anode.
lithium on the system, opo.
Can you talk a little about current developments? Dr. Torres said something about large-scale storage batteries. These are, of course, different from the batteries, lithium-ion batteries that you use in your equipment, although those could also be upscaled. For example, can you talk a little about flow batteries, redox flow batteries, which have a potential for essentially replacing peak generators, which are run on fossil fuels.
Sorry, I didn't get your question, sir. Can you repeat it again?
Can you expand the conversation to large-scale battery storage, which is, for example, the specialty of flow batteries, which have the capacity to replace peaking plants, which run on fossil fuels.
So for the fuel cell, sir, so it has a higher energy density compared to that of the battery. However, currently po, sir, me and my husband is also doing a research regarding the flow battery of zinc ion battery in collaboration with Chulalongkorn University. So we're just starting it with the zinc flow battery po.
And are there in Southeast Asia, are there examples of flow battery being now utilized to replace peaking plants?
Come again po, sir?
Are there now flow batteries that are being utilized to replace peaking plants?
Replace the
peaking plants, which run on fossil fuels.
Okay.
Well, essentially, this large-scale battery storage will replace run on fossil fuels, and some of the most likely replaceable plants are peaking plants. Plants that operate only when the demand exceeds supply and for a very short time, like an hour. So since you are working as well, and your colleagues are working as well on flow batteries, or distantly related to flow batteries, are there now instances where flow batteries are replacing peaking plants in our neck of the woods?
I haven't checked that one, sir, but I think there's already a vanadium flow battery that are being utilized.
But no, I mean expand, because you're in the renewable batteries space. May we expand the conversation beyond vanadium or zinc? Beyond vanadium or zinc to large-scale battery storage.
Yes,
to me, the most important, the most immediate application would be large-scale battery storage that will replace peaking plants that run on fossil fuels. Yeah, perhaps Dr. Fabella, may I expand a bit on your question? Uh, peaking, peaking power, you make use of it when the actual power is insufficient, right?
Yes, yes.
That's right. So the question therefore is, in her research or in her to her knowledge, are there technologies that are addressing this problem in our country? Okay. Now the other use of batteries, as I implied earlier, is to store uh, power or energy coming from seasonal resources like sunlight. So you don't probably need sunlight because you have enough power for that. But in the evening, you need battery to extend the time that your devices and whatever will need it. So on one hand, there is that issue on large capacity batteries, but on the other hand, because of the distributed factor, they don't really have to be large if you can distribute these smaller batteries to the different users all over the country. So that to me is one way of saying that this is the big problem in our country, and for our research to have impact, somehow they must connect to that problem.
Actually, Bill,
Mm.
actually, at the moment, you can utilize all idle rooftops of commercial establishments, and you don't need a battery. You are just going to be grid connected.
That's right.
So you will be able to reduce the demand from the grid for power. So
Right.
that is not necessarily battery based, right? But if you want to be completely off grid, if you don't want Meralco, then you will need battery. But you must remember that finance wise, once you install a battery with your solar panels, the cost easily goes double, and the payback period easily twice as much. So instead of payback period of eight years, you can have payback periods of 18 years. I'll probably not be alive anymore.
Right. Okay. Okay, but anyway, the issue we want Dr. Corpus to address is really whatever she's doing and what she and her husband will continue to do. We're looking forward to that impacting this needs, no, of humanity for storage power through batteries or as an backup for other storage generation, energy generation.
Have you had the big players in battery manufacture, renewable battery manufacturer, contact you or being interested in your work? In other words, these are the Teslas, the Panasonics, the Samsungs of this world. Have they made?
Currently po, sir, me and my husband started a startup company for the zinc ion battery here in the Philippines. So in addition to that one, another startup company is hired my husband as a consultant for the said battery type po, for the zinc ion battery here in the Philippines. It's located in Bulacan po.
Is this a multinational or a local company?
Local po.
Okay. I'd like to ask a question of Dr. Lantican.
Hello, sir.
Dr.
Morning.
Lantikan. Hi. You referred to harsh climate as the trigger for speciation, perhaps for the, no, sorry, Nantikan, right? For the development or speciation towards dwarfism. Am I correct? Did I hear you right when you said that in the past there was a harsh climate that triggered the development to dwarfism among coconuts?
Not necessary, sir. It's just the repeat elements or transposable or the jumping genes elements in the coconut genome that has expansions. So we cannot really say if it's due to the speciation or it is just an evolutionary or just a expansion in terms of genome. Because repeat amplification or proliferation has something to do with the gene regulation or the gene regulation of coconut as a whole. So it well, I cannot really say that it's that particular event has something to do with the development of dwarfism. It's just that that particular event or harsh event in the environment has something to do with the um, differences between the ancient species or the ancient coconut as compared to what we have now in terms of transposable repeats.
Dwarfs constitute a different species. They
No,
cannot.
sir. It's just a type of coconut dwarf. But based on our genome analysis, there's a certain event 400,000 years ago that triggered an expansion in terms of the dwarf or particularly maybe coconut as a whole. There's a particular event that amplified a certain genomic element in coconut, and that's the transposable element. And maybe it has something to do with or gene regulation of other um, genes for adaptation because there's a sudden change in the environment. So, but I cannot conclusively say that it's actually the cultivation of our dark coconut. No. Maybe the ancestor of that coconut, but speciation I cannot really say.
The problem with our coconut industry is that I think, and you mentioned it, it's very old trees.
Very old trees, sir. So
And, the term replant, replantation has always been there since maybe 50 years ago. And even without any new strain coming along, which is more productive, if you can just replant the old trees, you can just replace the old trees. Your production average will shoot up. The problem here is that we are not able to implement the replacement of the old trees for some reason. Well, maybe it's government that is
economic,
or maybe because over time we have subdivided plantations, coconut plantations, into smaller and smaller parcels, which means that the coconut farmer is no longer able to afford the investment, the capital cost, fixed capital cost of replant. So it is interesting that there could be new strains that are more productive. But I think in the Philippines, our biggest problem is our incapacity to use existing plant strains by simply replacing old strains.
Sir, sorry, there's an in terms of replanting, what we suggest, what the plant using the hybrids, because as compared to the previously standing plant, the hybrids has a higher heterosis or has a bit bigger. So ideally, this hybrid this can hybrids can have a higher yield as compared to the previously planted crop. The optimum, even it's old, the optimum yield the hybrids can still surpass that previously planted coconut seedlings in that particular area. So that's the advantage of hybrids using heterosis. Sorry.
No audio.
Thank you.
We can hear you, Dr. Villarreal. I think we can hear you.
Oh, you can hear me?
Yeah.
Okay. Earlier I was saying that I would like to congratulate all our presenters because they are already winners in their own right. Remember, you were 7, and we reduced the number to the finalists, and you are now 5. Having said that, I have 2 general comments, and I would like all the finalists to react in alphabetical order. All right, number 1, what will be your main take-home message to other young scientists like you? Because at the end of your presentation, you indicated a number of things that can be done, but what will be your main message? All right. Secondly, I would like to inquire also, what improvement will you make if you will retool the experiment that you conducted? I think that's all, and God bless all of you.
Hi, good morning, sir. For your first question, so for my message to the young researchers, it will be to be passionate about their work, and in order to help other people to help other people through researches, to be passionate. And then for the second question, it will be to improve my current research. I will be currently I'm doing a low energy current density, so I will be jumping into a higher energy density to address more issues.
Thank you.
For the young scientists out there, maybe my take-home message would be, just don't be afraid. We are all good. Young scientists or Filipino scientists are all good. We just need to be creative enough and do everything, do your work with the heart. So that's my take-home message. And for the improvement of our current methodology, maybe because my current assembly is still fragmented. So what I want in the next research would be assemble the entire chromosome of coconut, not just genomic scaffold, but the entire chromosome itself.
Please introduce yourself when you speak, because you have the mask. We cannot recognize you.
I'm Darlo Lapitan.
Okay, second presenter.
Good morning, I am Lorico Lapitan, sir. Lorico Lapitan, I'm the second presenter. And my message for young scientists and aspiring scientists is that science is great, okay? Science is great, and our country needs scientists for nation building. Okay, a lot of countries in our Southeast Asian neighbors, they are actually having already in the tiger economy because they're investing a lot, their governments are investing a lot in science. Now there should be acres of this science field. And my suggestion is, okay, do not be afraid, science is great, and be a part of nation building. Okay, if I will answer the second question, what part of the methodology I will change, I think I will actually add more samples, okay, and look at other, let's say, mutations that are linked to cancer, something like that. Thank you very much.
Thank you.
Hi, sir. I'm Zomesh Maini from Ateneo. So, my first, to answer the first question, if I had a message to younger scientists or people who would really want to get into a career in science, it's probably first, you don't have to have the best grades at the start when you're going to want to get into science, because sometimes a lot of people are discouraged very early on simply because they feel that they don't have the necessary aptitude or the necessary grades, or they had science experience in their life. But then the curiosity is there and the love for knowledge is there. So don't let that discourage you, because if you try to become a scientist or a scientist in training, I haven't completed my PhD yet, so I'm still studying. You will continue to try and learn for the rest of your life. So there's always going to be an opportunity for learning. And sometimes you're going to learn from people who are much younger than you, and that's okay. And you have to really just keep on moving forward. So that's the first, to answer the first question. The second question, what would I do to improve this work? Actually, this isn't really my dissertation topic. I have a different dissertation topic. This is a project that we did. But if I was going to improve it, I would probably add three things. So the first would be to add an SEMEDX study on the fungal biomass in order to be able to quantitate the functional groups to which the heavy metals bind to, so that we can really explain how they do it. And then the second one would probably be to also take a look at a simple transcriptomic study that would compare unexposed and exposed versions of the highest performing fungi, so that we can see which genes are turned on and off, for example, increased or depressed, in response to the increasing exposure to heavy metal concentration. And probably the third, which would be the most simple among all of them, is to try batch scale studies utilizing the fungal material as a biosorbent, so that we can have something that is actually industrially applicable. That's it, sir. Thank you.
Thank you.
Good morning, sir. I'm Buford Johnson Cañas from the UP Marine Science Institute. So, for perhaps to answer the first question, to encourage, as an encouragement to the young scientists, I'd like to say that there is a lot of opportunities for us to really contribute to the science and advancement of science and technology in the Philippines, and that we just need to cultivate the passion that we have. And yeah, I mean, there's a lot of opportunities out there as to where we can contribute. And for the other question, which I'd like to improve on my research, I'd very much be pleased if I would have wider scale sampling. So my sampling is just focused in the Indo-Pacific primarily, and have like several samples from the Indian Ocean and just very few from the Atlantic Ocean. So in order for me to like to really look into the diversity of hydrocrasters, I would want to be able to have samples from several of these areas.
Okay. May I ask a question?
Yes, academician.
Okay. I'd like to ask this question to Mr. Lapitan because the detection that you indicated is an early detection of cancer, right?
Yes, sir.
I wanted to know if there is an analogy between what is happening now in the detection of the coronavirus. There is that effort at ito yung testing, di ba?
Yes, sir.
But there is another form of testing which detects much earlier than when the virus enters your body from somebody else, right?
Yes, sir.
Is there any relationship between the two, or is there not, but an analogy of what you have done to this mechanism that I don't even remember the name, but I just have that information that,
The sandwich sensor.
No. This in the coronavirus thing, there is trying to test your blood, right? To know the level of the white cells, I think, or something.
The
Oh my
antibody,
God.
sir,
Right.
okay. So basically, sir, I
So
think
it's an early test, right, which is easier to do, and you can do more of. Is there any analogy you can?
okay, sir. So to answer your question, we have two types of tests for coronavirus. The one that we say, the one that's the fastest test is called the antibody testing. It
Right.
actually tries to identify any antibodies, for example, that your body was able to produce to combat the virus. Okay, but these are rapid tests. Okay, and obviously, since they are rapid tests, we can do a lot of tests at one day, as compared to the other type of testing, which is the PCR testing, which would take what's this more time to finish, something like that. Now, I think the drawback for the antibody testing is that it is prone actually to false positives. Okay, so for example, one of my friend got positive in the antibody testing, and then he was subjected to a swab test or PCR test, and it came up negative, something like that. So we have to be careful as well with the data that we are actually receiving from these two types of testing. Okay, now to answer your second, the other question, if there is actually a link of what I did, is that right, sir? Of
Yeah.
what I did to coronavirus testing? Yes. Okay. So basically, since as I've mentioned in my presentation, okay, so I have developed this sequencing strategy that can detect a cancer-related DNA down to, let's say, 1.6 attomolar. Okay, so even at low concentrations of this DNA, we can already detect it, something like that, without the use of PCR, which is expensive, and not all laboratories are equipped with PCR in the Philippines. Okay, so I think the method, okay, that I developed can be easily translated to, let's say, testing the viral DNA. Of course, obviously, we have to transform the RNA from the virus into a DNA, something like that. My study will be easily translated to the testing.
Thank you, Miss.
Thank you, sir.
Thank you.
Okay, so
And
questions from academician Gisela Concepcion. Ma'am?
So I'd like to applaud each finalist for your impressive work and presentation. Each of you deserves high recognition. All studies have nationally relevant and wide applications, though some are at the early stage and others are in the more advanced stage. But for purposes of this competition, I would like to give each equal time and opportunity to answer these questions. First question, which I know academician Joey Balmaceda is also asking, is which part of the work you presented were you directly involved in? The second question is partly answered already with academician Villarreal's question on improvement, but phrased another way, what unanswered questions or unsatisfactory or limited results did you have that you would pursue in the near future? Okay, and this you would answer with humility. Now, in particular, in the case of Dr. Dr. Lorica, I'd like to know whether the SNP, the cancer SNP that you are detecting is an indication of cancer or predisposition to cancer. In the case of Dr. Lantican, I'd like to know whether you will pursue the particular enzymes that are related to the biosynthesis of high-value triglycerides, monoglycerides that
coconut oil is known for. That's it. Thank you.
All right. Thank you, Ma'am Giselle, for that question. So, to answer the first question, I'm essentially all of the in my work from the I wasn't really involved in much of the collection efforts as many of my samples came from around Japan and around Indo-Pacific areas. So most of these samples were actually sent to me by my collaborators, and from where I did all the DNA extraction, sequencing, and then molecular phylogenetic analysis, as well as the morphochemical observation. So essentially, aside from the collection of samples of hydroclastus from around the world, essentially most of those in all of those steps, I am the one who is really working on my research, because well, it's essentially my research. So number two is what unanswered questions. For me, one of the problems that we have is that while I looked at the biodiversity and systematics of the family Scythocyphonaceae, and most of the studies were actually conducted in cold water species, and we are not yet and we have not yet done a lot of work on tropical species. So what I would want to clarify for my future research, and which I am actually also in trying to work now at PPMSI, is to look into the diversity and as well as the molecular phylogeny of this not just brown species, but um, uh, molecular phylogeny of the members of the family Scythocyphonaceae, not only in the Philippines but also in other areas of the world. So I am still collaborating with some of my colleagues from different areas uh, uh, within the tropic. So as of now, we are still pursuing that kind of research to answer the questions on biodiversity and systematics of brown seeds, in particular the family Scythocyphonaceae.
Hi, ma'am. So I'm Mr. Maini from Ateneo. So to answer your question number one, well, basically I was involved in everything except the inductively coupled plasma emission test, because the inductively coupled plasma test was performed through the Philippine Institute of Pure and Applied Chemistry. It was outsourced because number one, we don't have that machine, and number two, it was actually a good practice. It was actually good practice because it was a third party, so they didn't really know what they were measuring. So they gave us really good accurate measurements of the values of the work. But then everything else, the fungal culture, the collection, the subculturing, the computation, even the writing of the paper. So I wrote that paper. Um, and the reason why I was involved in all of the steps was because if you remember, I mentioned that the co-authors for that paper are undergrad students. So of course I have to teach them how to do everything, and I have to be there when they're doing everything and do it together with them as well. So it was actually a product of teaching as well, this particular paper. Then to answer your question number two, what unanswered questions or unsatisfactory limited results? I think the majority of the paper was really presenting new records and new abilities of fungi that are currently known but really unexplored, because no one's really looking at them. But if I was going to develop that further, I would really go for the transcriptomic work that I was talking about earlier, because it's not enough to say that this is what is. We need to be able to explain how. And it might have implications on the way heavy metals are carried as well into human cells, because fungi are eukaryotic. And the most prevalent theory of how they are able to accumulate heavy metals is that they use already existing transporters to transport this, which may be routes for the heavy metal lead to enter human body systems, because that particular aspect is not really understood as well currently. Thank you.
Hi, ma'am. Good afternoon po. For the first question, I basically involved in every steps in the bioinformatics analysis. The only thing that I'm not involved with would be the next generation sequencing because we just sent the DNA for outsourced sequencing. So for the second question, the unsatisfactory or limited results, basically coconut genome or the genome sequencing is just a prediction. So it always a subject of further validation. For example, you can perform transcriptomics, you can perform genome editing, you can perform genetic engineering in order to validate the function of all the genes in coconut. But then having it predicted makes it a better reference in order to perform, or it's a requirement in order to perform these validation steps. Now, for the question about the oil genes, yes, we can have further functional validation, but the functional validation step would be the isolation of the actual protein and validation it using in vitro assay. So in order to fully validate that this particular allele or particular SNP is really associated to the increase in oil production.
Hello, ma'am. So good morning. So to answer your first question po, in what aspects of it that I was involved in the study? So basically, my study has, let's say, three parts. Okay, the first one was material synthesis, which involves the use of magnetic nanoparticles and the synthesis of the polymer, and obviously the use of the DNA probes. Okay, the second part of the study involves the biosensing part, and the third part involves the designing the polymer bead amplifier. So basically, ma'am, for parts 2 and 3, I was the one who designed this. Okay, but in number 1, wherein we have just decided, ma'am, to buy, let's say, the magnetic nanoparticles and to send the samples of the DNA sequences, which we also designed something like that. This is because the main focus of my study that time was more of the biosensing. So, of course, as a PhD student, you also have a limited time doing work, so you have to really look at which aspect in your PhD should you really be working on. Okay. Now, for the second part of the second question, I think, ma'am, if I will do, not really unsatisfactory results, maybe, let's say, I should have used, let's say, or um, like pure plasma samples from patients, because in my study I've used I did biosensing in phosphate buffered saline, and in phosphate buffered saline spiked with 10% human plasma. That is just to mimic clinical assay conditions. But it would have been great, for example, if I use a pure plasma, something like that, to dissolve my analyte or my target DNA. Now, to answer your third question, I did some literature reviews, okay, to decide why, let's say, I use this specific, let's say, SNPs. And based from what my findings are and from the studies of students who did previously, okay, in the lab, they saw that this specific SNPs that I use in the study are actually coming from sequences of samples which are positive for cancer cases po. Thank you po.
Hello po, ma'am. Good morning po. So, I'm Lynn Marie Corpus po. So, to answer your first question, I did everything po from the material synthesis to the materials characterization, analysis, as well as that of the battery characterizations for the three types of batteries, for the lithium-ion battery, sodium-ion battery, as well as that of the zinc-ion battery. The only exception is that for the sodium-ion battery, for the sodium-ion battery testing, it was sent to Taiwan since they already have the materials and equipment to fabricate a sodium-ion battery. However, for the other types, I already did everything. And then to answer your second question po, which part I'm not satisfied or I want to improve, it will be since for all my batteries, I only run it until 200 cycles. To see until which cycle it will fail. So the maximum number of cycles my battery can operate. So I would like to check that one since during my PhD studies, the 200 cycles already took me 2 to 3 months to finish due to very slow charge and discharge cycle. So if I'm going to wait for it to fail, so it would take me a lot of time. So I would like to check that one first po. Thank you.
Okay, so I think we're down to at least one last question, and this is question from academician Balmaceda to Dr. Corpus. So does the introduction of vanadium oxide and nanomaterial improve the safety feature of the batteries, particularly CIV?
For the ZIB, thank you for question po. For the ZIB, there are only three cathodes that are being considered, such as the manganese dioxide, vanadium oxide, as well as the Prussian blue. So among these three cathode materials, only the vanadium oxide have the higher capacity compared to that of the manganese dioxide and the Prussian blue. So basically, it greatly improves the electrochemical performance of that of the zinc ion battery in terms of its specific capacities po.
So we have a question from the DOST Undersecretary, Dr. Solidum.
Yes, thank you. You have presented many applications of your research. Can you just repeat if you have not, but I think you have mentioned this already, but what is the most important application? How far are you from realizing this application? And what do you need from the Department of Science and Technology to support the application? Thank you. That's my question for all the 5 presenters. Congratulations, by the way.
Okay, so the most important part is the application on the zinc battery. So zinc ion battery does not need any vacuum technology or specifically the glove box or to isolate the system from air to manufacture and to fabricate the zinc ion battery. So basically, it can be easily be fabricated in the Philippines without any sophisticated equipments. So in addition, zinc ion battery is very stable upon contact with air and moisture. So we can easily use water as electrolyte and does not need any organic or ionic liquid for the solvents. So what I need from the department, from the DOST is to funding from my research to continue what I have started and to improve or to develop what I have started po. And also, in addition, I have started po a startup company. So I hope the DOST can also fund the startup company with respect to the improvement of the zinc ion battery here in the Philippines po. Thank you.
Basically, what we provided in this research is the availability of molecular marker resource for improvement of coconut breeding programs we have in the Philippines. So without, of course, DOST, we expect more funding, of course, because since it's DOST who funded this research, and we want it to continue. And by this time, I really want to sequence not just one variety of coconut, but other varieties of coconut in order to increase the efficiency of plant varietal improvement programs. So that would.
Thank you, sir. Perhaps my research is really basic in the sense that I am describing biodiversity. So in terms of application, perhaps the contribution of this research is that we actually provided also DNA markers that we can use to consistently identify and correctly identify the seaweed resource that we can tap for development. So as I've mentioned in my presentation, hydrochlorus is currently they possess high-value natural products and also bioactive compounds that can be tapped for medicinal and other applications. So these DNA barcodes can also be used to consistently identify those seaweeds. And with respect to the funding opportunities, I've also mentioned this in my presentation that we really need to boost our support for seaweed biodiversity research because these are foundational knowledge that we would want to generate in order for us to actually develop our seaweed resources. Because I mean, basic research, particularly biodiversity and their distribution, is very crucial in order for us to identify, I mean, what we have, where we have them, and when we can have them, so that we can actually uh, push forward towards the development of these resources for uh, improvement of lives, especially of our coastal communities.
Hi, sir. So I mentioned towards the end of my present. So I am the one who presented about fungi. So I mentioned towards the end of my presentation that the use of living cells was not so good for industrial application. And we actually have a second paper where we developed a biosorbent that can remove 99.5% of lead from a solution, and it's not alive. So it's made of immobilized fungal material. And while that paper, the results were quite successful because it was also proven to be reusable, we did it using small scale versions. So I think in order to really improve that and make it translatable into something that's commercially and industrially useful, we need to make big batch biosorption experiments. And maybe if we were trying to do that, we would probably need, of course, apart from the funding, the space to actually test it. And we're dealing with heavy metals as well. Ateneo is a smaller university compared to a lot of others. So if mahirap po sigurong gawin yung malaking large scale dealing with heavy metals because we have to be mindful also of how we handle them and how we dispose. So that would be of great help if there's a space where things like that could be done. Thank you very much.
Thank you.
Hello, sir. Good afternoon. Morning. So I was the second speaker a while ago, and in my presentation, I've emphasized, okay, early diagnosis is very important for us to pick up occurrence of illnesses or diseases. Okay, so the application actually of my study is actually in biodiagnostics. Okay, so obviously, I think everyone would agree, okay, on the importance of early diagnosis because this will help, for example, to curb the spread of diseases. Just like if we can flatten the curve, that's very nice. Okay, and we can with early diagnosis we can also improve the lives of people and obviously save our economy from, let's say, an impending economic doom, something like that. Okay, so just to answer the second question, like what do we want from help, like form of help from the Department of Science and Technology, I think all my other four mates here, okay, do agree of funding, but I would just like to emphasize as well that DOST should also continue in giving scholarships, let's say for masters and for PhD students. I mean, DOST should actually continue in giving these types of scholarships because we need warm bodies, okay? Because the thing is, we at my level, I try to mentor students, something like that. So even if I have a funding, but if there's no one to mentor, that is also becomes dysfunctional. So I think the two things that we have to realize is obviously the funding from the government, and of course the continuous help of the government to support scholarships in the masters in the PhD levels. Thank you very much, sir.
My question has to do with all of us, no, in the science and technology and engineering and innovation. Okay, it has to do with the entire community of science and technology stakeholders. So my question, which you could answer very simply, what is it that we need to do as a community that we will do ourselves without having to ask for help from government or from big companies? What is it that we have to do as a community to enable us to move our community to upgrade our capability to do more science and technology in the long run? Thank you.
Thank you for that question, sir. So I think for me, what what the science, technology, and innovation community is actually lacking at the moment is that we are really challenged in our capacities to actually communicate our researches to the wider population. And that is also, I mean, that puts us in a disadvantaged position because, I mean, if we can communicate actually our science and the technology that we have, I believe that the society would actually pour their support and will also rally behind our backs as we pursue science, technology, and development in the country. So
Thank
thank
you.
you.
Hi, sir. So, good morning. To answer your question, I was able to attend a talk of the DOST secretary when he visited the Philippine Institute of Pure and Applied Chemistry. And he mentioned that one of the major problems of the different institutes under DOST, for example, was the utilization rate of the different researches that were being produced, irregardless of whether it comes from whichever university or institution. So I think as a community, what we can do really is to enter into conversations that align the efforts or maybe the similar efforts of people coming from different places towards the public agenda and the national agenda as well. So there has to be a connection with whatever the national goals are and whatever the public needs are in terms of the research that's being pursued. Sometimes I think, based on my own experience and my experience of interacting with other people, we center around research that we're familiar with simply because we don't really enter a lot of conversations with other people from other places. And because of that, I think that severely limits the ability of the community. Only if people maybe would talk a little bit more and enter more collaborations and streamline their efforts, then maybe we can move a little bit better forward. At least in my opinion, that's something that I see that we should be doing. Thank
Thank
you.
you.
For me, let's take advantage of the existing, but not now we are only working from home and take it as advantage into information dissemination. We know that science and technology is a bit hard to understand. Uh, some of the terms are not fully understood by the local community, but we can try to create some infographics or make use of the internet platforms in order to make it to simplify the product of science and technology. And that I think would be the great what I think is a great contribution in order to further increase the appreciation of all our countrymen and pursue careers in sciences and technology.
Hello, sir. Good morning. So I'm just speaking based on my experience. So I consider myself to be a material scientist, dealing with more on bioanalytical sciences. And sometimes and most of the time in my studies, I communicate, I collaborate with microbiologists, with biologists, okay, with synthetic organic chemists, okay. And I think this is one thing that we should actually, push through in our scientific community, and that is multidisciplinary research. Okay, because obviously science and technology research nowadays is no longer traditional. It involves multidisciplinary research. Okay, so I think it would also be nice, aside from multidisciplinary collaboration, we can also, as a community, share our own expertise through webinars, through talks, something like that. And I think those are two important, ways in which we can actually move forward as a scientific community. Thank you, sir.
Good morning po, sir. So to answer for your question, I think it will be better to start with small children to explain to them or to show them some small scale experiments or homemade experiments to inspire them to pursue and to continue with that of the science and technology courses or in courses and to do some experiments. But I think at an early stage they can already appreciate small scale experiment that can inspire them, and at the same time they will learn they will learn on the process. It's much easier to learn when you actually observe the experiment rather than listening or just viewing it. So I think it will be better to start a small scale or homemade experiment first for small children, and then going to school to start a higher type of experiment. Then it will inspire the community to pursue the more science and technology courses po. Thank you.
Okay, I want to thank everybody for their very good response to my last question. And this is by way of saying that I think you have really not only accomplished the things that you did in your respective research, but you also realize the responsibility as members of this community that we need to help each other. And why not do this in a big scale, like for the entire science and technology community in our country. Again, thank you very much.
Okay, may I ask the panel of judges if they have more questions? Academician Villarreal.
Okay, well, this is not really a question to the finalists, but rather a reaction to the comment of national scientists Pavella with respect to replanting of coconut. I agree completely that if only you replant with newer planting materials, the yield of coconut will be really improved greatly. But I would like to inform the group that the Coalition for Agriculture Modernization in the Philippines, headed by national scientists here, is deeply involved with the Philippine Coconut Authority in the massive replanting of hybrids in Quezon Province as the first province. It will later be done also in the Bicol region. So with that activity, we believe that there will be tremendous improvement in the so-called yield of coconut in the Philippines. But the study of marlon is a very significant development as far as the coconut industry is concerned, and we hope that that particular study will be continued and fully funded by the Department of Science and Technology. That's all, and again, congratulations to all of you.
Okay, thank you, Academician Villarreal. So I think National Scientist Pavella was raising his hand earlier. Sir?
Perhaps it's time to wind this up, but before that, I'd like to ask a very simple question. Anybody can answer it. As a budding scientist, do you see yourself as in search of the truth? And will you ever get there? You can just simply say yes and yes, yes and no, no and yes.
Sir, can you repeat your question?
As budding scientists, do you see yourselves as in search of the truth, which means that your holy grail is the truth? And will you ever get there?
So he was asking if, as a budding scientist, are you for the truth?
Do you see yourself as in an enterprise in search of the truth? And will you ever get there?
For the first
there's no real correct answer here. It's perspective.
Yeah, so the first question is a yes, and for the second question is hopefully.
To answer your questions, for the first question, yes, because the practice of science is built on trying to understand the truth, so yes. But for the second question, probably no as well. I mean, there's always a limit to what we can describe using science, so there will always be something left that we can't really understand completely.
For the first question, sir, I think yes, I'm in search for the truth. And for the second question, maybe because I'm looking into DNA and we are not yet, the science is actually evolving. So there's a lot of possibilities to enter.
For the first question, sir, it is yes. And then for the second question, hopefully yes. Though there are a lot of uncertainties, but I hope that we can still find the truth within those, or at least partial of those truths we can already acquire or determine po.
Hi, sir. Good morning. Okay, so to answer your first question, yes, we have, as a scientist, we have obviously we have ethics, and we should always be for the truth, something like that. Now, for your second question, obviously, I am still on the journey towards the truth. Thank you.
Okay, just food for thought. My own answer to the two questions is no and no. You are not in an enterprise in search of the truth. You are in an enterprise in search of a better description of reality. You will never get to the truth because there's no such thing as truth for lab rats of science. There is only a better description of reality. And a better description of reality today will probably be replaced tomorrow because all the canons of science are provisional. Provisional. In other words, they are not like mathematical actions. They are not like mathematical theorems. They are not like Pythagorean theorems, which were true in the time of Aristotle and are still true today. But science is always a provisional enterprise. That's only food for thought. You may agree with me, you may not. Thank you.
Thank you, sir.
So,
yes, sir.
okay,
yes, sir.
so maybe we should wind this up. And winding up means that our participants will no longer participate in the deliberation. They'll probably have to move to another room while we do the deliberation. So thank you very much. It's been very enlightening. It's nice that these budding scientists are enlightening the oldies and old faggots like myself. Please continue. And I am personally hoping that one of you guys will bag the biggest prize in the science world one of these days. Thank you.
Thank you, sir, and noted. So at this point, I would like to reintroduce the 5 finalists. So right here with me, camera please. Please. Para makita kami. So this right here with me is Dr. Wilfred Jan E. Santañez, and he is from the Marine Science Institute of the University of the Philippines in Diliman. And here, okay, so Mr. Somesh Artus and Maini, and he is from the Ateneo de Manila University, Department of Biology. And right here on the middle, we have Mr. Darlon Villantican, and he is from the Department of Agriculture, Natural Sciences, College of Arts and Sciences from the University of the Philippines, Los Baños, in Laguna. And here, well, actually, he was my student. Right here is Dr. Lorico de los Santos Lapitan Jr., and he represents the Department of Chemical Engineering, University of Santo Tomas. And last but not the least is the talented finalist, again from the Department of Chemical Engineering from the University of Santo Tomas, Dr. Lynn Marie de Juan Corpus. Let us give a round of applause for all our finalists. Okay. So we're down to the last part of our program, and we shall now hear from the DOST Undersecretary for Scientific and Technical Services, Undersecretary Renato U. Solidum, for the closing remarks.
On behalf of the Academy and the Department of Science and Technology, I would like to congratulate all the finalists for a job well done, especially for their excellent paper presentations. I commend every one of you for showcasing your contributions that can help our country address various problems that beset our society. And I would like to encourage you to continue to be inspired to do more researches for our country's benefit and for the benefit, of course, of our society in general. I am one with the National Academy of Science and Technology in encouraging our talented scientists and young scientists, of course, and researchers by recognizing their efforts to advance the society through science and technology. The Department of Science and Technology has always been passionate in supporting this endeavor through giving of research grants to the winners of selected NAST awards. So in behalf of the Academy and of course the Department of Science and Technology, I would like to also take this opportunity to thank every members of the Board of Judges and to our chair for the diligence and consideration in seeking the best talents among the roster of the aspirants for this year's award. Furthermore, my sincerest appreciation to the NAST community headed by academician Fedora Asansa for inviting the Department of Science and Technology to be part of this very important activity. Thank you very much to everyone. Good day and continue to be safe. Mabuhay po kayong lahat.
Thank you, Undersecretary Solidum. Again, my congratulations to the 5 finalists for making this far. Like National Scientist Fabella has said, you are already winners for having chosen from the pool of nominees we have received this year. Thank you everyone for joining this activity via Zoom or Facebook. The results of this exercise will be announced the soonest possible time. The awarding ceremony will be in the afternoon of July 10 via Zoom as part of the NAST 42nd Annual Scientific Meeting slated on July 8 to 10. We hope you could join in this very important annual activity of the Academy. Once again, thank you very much. We will now be signing off.